welcome. I'm going to talk about inherited variations in drug and neurotransmitter metabolism. And as you know from the invitation, Professor Weinshilboum got his education at University of Kansas, and then he trained at Harvard, and with Axel Rod at National Institute of Mental Health in Bethesda. And Dr. Weinshilboum is here to give this lecture. He also was opponent on a thesis here, this catecholamine O-methyl transferase activity in human erythrocytes, which has been performed in the basement here, this building, and was defended Monday in Uppsala. This is together with Department of Medical Genetics in Uppsala, and this also show that Dr. Weinshilboum is a fellow of many talents. He is a clinical pharmacologist, psychiatrist, geneticist, and he has the William McKnight 3M Professor of Neuroscience at the Mayo Medical School at the Mayo Clinic. And you're very welcome to Stockholm and to tell us about the inherited variation in drug and neurotransmitter metabolism.
Thank you, Leonard. Dr. Vetterberg's introduction, I'm afraid, sounds like the old American and English saying, jack of all trades and master of none. We'll see before we're done here. Let me say first, what a great pleasure it is for me to be here today to share some of the work which many people in my laboratory have been involved with over the years. And I want to make that point initially, and then before we're done, I'll even tell you who they are. What I'd like to do in the very short period of time that we have this afternoon is to briefly review some of the work which we've been doing, looking at the possible role of inheritance in the regulation of variation of the function of one relatively important but small part of the nervous system, namely the adrenergic neuron, both in the central nervous system and in the periphery. And as I don't need to tell anyone in this room, clearly it is thought that the adrenergic neuron plays an important role in a variety of disease processes, or MAY, diseases such as the affective disorders, schizophrenia, and even diseases that some of us as internists are interested in, such as essential hypertension. In addition, drugs which affect the adrenergic nervous system are clearly used to treat all of these diseases. We also know that all of the diseases that I've mentioned have a high degree of heritability. It appears that genetic factors may play a role in the pathophysiology of these diseases. And what I'm going to do is to describe to you one approach, and only one approach, to beginning to try and understand the role of inheritance in the regulation of individual variation in the function of this one neurotransmitter system, which may play a role in a variety of disease processes. Now, if I can remember how to get all the wires turned on, maybe we can. I can't believe it, it actually works. Before we begin to talk about the role of inherited variation in the regulation of this important part of the nervous system, let's briefly review the function of the adrenergic neuron, which is basically the same both in the central nervous system and in the periphery. Here we have a green schematic representation of an adrenergic nerve terminal in which the neurotransmitter, and that could be either norepinephrine, as we call it in the United States, or noradrenaline, as it's called in Europe, or dopamine, is formed in a biosynthetic sequence, stored within vesicular storage sites, released to the exterior of the cell in response to nerve stimulation by a process of exocytosis, interacts with receptors found in effector cells, and then has its actions terminated primarily by uptake back into the nerve by a specific neural membrane uptake mechanism, or by metabolism by the enzymes catecholamine transferase or monoamine oxidase, either in the immediate area of the synapse or in peripheral locations such as the liver or the kidney. Now, what we're going to do today is to focus our attention on the only one part of the functioning of the adrenergic nerve terminal in this entire neurotransmitter system, and that is metabolism. We could equally well be talking about variation in function at a variety of these other steps, such as biosynthesis or the neural membrane uptake mechanism. But let's focus our attention for the time being on the enzymes that are involved in the metabolism of neurotransmitters. And let's take a neurotransmitter that psychiatrists love to talk about, dopamine. This is a catecholamine neurotransmitter which can be metabolized by the enzyme monoamine oxidase to form an oxidized product, by the enzyme catechol-O-methyl-transferase to form an O-methylated product, or, as we're becoming more aware, by forming sulfate conjugates via the actions of an enzyme phenol-sulfotransferase. And I think the important point to be made here is this, that there isn't just one pathway for the metabolism of catecholamines, but there are a variety of pathways operating in parallel, potentially operating in parallel. And it might well be that important changes in behavior could be associated with not changes in just one, but changes in many of these enzymes. And we'll come back to that theme before we're done with our discussion today. But for the time being, we're going to focus our attention on just one of these enzymes, and that will be catechol-O-methyl-transferase. And let's see if I can, there we go. Now, the problem that those of us who are interested in the function of the brain in intact, unhomogenized human beings have is this. We don't have people who are willing to give us brain samples to study their enzymes, and rightly so. If we want to begin to look at, to gain insights into the functions of these enzyme systems, then we are stuck in man with attempting to find some indirect method for assessment of what's going on where we're most interested, namely in the nervous system. And one way, and only one way to do this, is to begin to see whether the enzymes of interest are present in an easily accessible peripheral tissue. And for practical purposes, that really means in blood elements or perhaps in skin fibroblasts. We do that without making any assumptions that either the biochemical characteristics or the regulation of the enzyme measured in that accessible tissue is any reflection of what's going on in the brain. We don't jump to that conclusion. That's a hypothesis which must be tested. In order to test that hypothesis, you first must have the enzyme available in an accessible tissue, and not all of the catecholamine enzymes are. You must then develop an accurate assay to measure the enzyme activity, and in many cases, although that looks like that ought to be easy, that happens to be one of the most difficult parts of this procedure. If you can do those two things, you then want to determine what the population variation is. Are people all very similar, or are they somewhat different with regard to the regulation of the enzyme activity? If there is a variance, you would like to know whether that variance is under what the regulation is. That is, do genetic factors play an important role? Does environment play an important role here? And assuming that you can do all of these things, does it really make any difference in terms of other tissues? After all, a red blood cell is not a neuron. And so you want to know whether what you're looking at in a red blood cell or a platelet bears any relationship to the enzyme activity in other tissues, such as the liver, the brain, the kidney, in which you might have greater interest. And finally, does that have any functional significance? Does it make any difference in terms of individual variations in the metabolism of neurotransmitters or individual variations in the metabolism of drugs related to other neurotransmitters? And I could just as easily label this slide variants of human drug metabolizing enzymes, because most of these metabolic enzymes that I've talked about are not only involved in the metabolism of neurotransmitters, but of a variety of drugs which we use that are related to the neurotransmitters. Catechol-O-methyl-transferase is involved in the metabolism of L-DOPA, used to treat Parkinson's disease. It's involved in the metabolism of alpha-methyl-DOPA, used to treat 5 million hypertensive patients in the United States alone. It's involved in the metabolism of isoproterenol, used to treat millions of asthmatic patients. And finally, I think it is probably foolhardy to look at one of these enzymes alone without thinking of the effects of the other enzymes. And let me give you an example involving a drug that we could just as easily use a neurotransmitter to show. That is, here's the drug L-DOPA, which is used in the treatment of Parkinson's disease. We know, as I mentioned a moment ago, that catechol-O-methyltransferase is involved in the metabolism of this drug, but also the sulfate conjugating enzyme that I mentioned, phenol sulfotransferase, is involved in the metabolism of L-DOPA, and so is the enzyme dopa decarboxylase, which is, after all, the reason we give L-DOPA, so it will be converted into dopamine in the brain of these Parkinsonian patients. It might be that variation in this enzyme will only become important in certain situations in which this enzyme activity is also changed. Now, I said that we were going to focus our attention initially on the enzyme catechol-O-methyltransferase. So here's that same slide we saw before, showing that catechol-O-methyltransferase is found not in the nerve terminal, but in effector cells in the immediate area of the synapse. It's found in high concentrations in the liver and kidney, and it's found, interestingly enough, in the erythrocyte, in the red blood cell. And that was described by Axelrod and Cohn and many investigators around a decade ago. What we're going to begin by doing is going through this stepwise sequence that I just outlined for you, looking at the possible role of individual variation in this enzyme activity as a reflection of what might be going on elsewhere, and determine whether that might have any functional significance. Now, I want to begin immediately by saying, please, I don't want someone to immediately say, there was this crazy fellow who came from Minnesota who said if you stick a needle in somebody's vein, you can tell what is going on with an enzyme in their brain. That's the hypothesis that's being tested here, and we'll see whether or not that bears any relationship to the truth before we're done. So let's focus our attention on catechol-O-methyltransferase, and what happened really went something like this in terms of these studies. Here's the reaction that catechol-O-methyltransferase catalyzes. The most famous of the reactions, norepinephrine or noradrenaline, is converted in the presence of magnesium to a ring O-methylated compound, normetanephrine, with S-adenosylmethionine as a methyl donor. Now, about 1973 or 4, we were involved in studies of genetic regulation of one of the catecholamine biosynthetic enzymes, dopamine beta hydroxylase. And that involved my getting up at 6:30 in the morning every day and going to every school in Rochester, Minnesota, and getting blood samples on 30 children, which were all we could process, and bringing them back and centrifuging them. And I was using the plasma to measure the dopamine beta hydroxylase activity, and we were throwing the erythrocytes away. Well, since I was getting up that early every morning and going through the snow, and I think we have as much snow in Minnesota as you have here, it broke my heart to throw those red cells away. And so I came into the lab and told my technician, Mr. Fred Raymond, I said, Fred, we're not going to throw those red cells away anymore. In 1970-71, Axelrod said that catechol-O-methyltransferase is present in the red cell. We're going to do a very simple experiment. It'll just take a couple of weeks. Believe me, this will be an easy one. We're going to set up that assay for COMT, and we're going to see whether brothers and sisters are closer than unrelated people with regard to the level of erythrocyte COMT. We're going to see whether there's any familial aggregation of the level of this enzyme activity in the red cell. It'll only take a couple dozen samples and we'll be done. That's what I said in 1973. This shows you the assay that we ended up using, by the way, using 3,4-dihydroxybenzoic acid, a catechol, which gives you relatively the same activity as the normal neurotransmitter, and we use a radioactively labeled methyl group to result in the formation of this radioactively labeled product, which we can extract and measure, and that's the way we measure the enzyme activity. But when we tried to set up the assay that Dr. Axelrod described in 1970-71, we had all kinds of troubles. That's why line 2 on that slide said, accurate assay. We couldn't get reproducible results. We got all sorts of variation. We had white precipitates forming in our test tubes, and I called Dr. Axelrod up, and it immediately became clear that he had a phosphate buffer in a tube to which he was adding magnesium with lysates that contained calcium, and we were getting calcium, magnesium, phosphate precipitates. The pH was changing. All sorts of bad things were happening in the test tubes. And I said, Julie, didn't you notice that white stuff in the tubes? And he said, sure I did, Dick, but the basic point was the enzyme activity was present in the red cells, and that's all we wanted to say, and I'll let you work out the details. Now, as a matter of fact, he's always right. The man is always right, and he always leaves it for folks like me to work out the details, which is just fine. And it took us about 2 years to work out the details and get an accurate assay. And I only show you this to show you that what we did was we used purified enzyme. We had to look at the effects of calcium and magnesium on the purified enzyme. We made the observation that calcium, which could contaminate these blood samples, was a potent inhibitor of COMT. Here you see a sample. I'm showing you this with the red cell enzyme, but we did exactly the same thing with the purified rat liver enzyme. If you remove all calcium and magnesium and then add back magnesium to activate the enzyme, calcium will inhibit. Magnesium and barium have much lesser effects, so we had to get rid of that bad calcium that might be contaminating our samples. And the assay we eventually came up with after about 18 months was to take the blood, lyse it in water, add solid beads of Chelex 100, which is just a bead with EDTA covalently bound, shake it up. The bead gloms onto magnesium and calcium. You can then centrifuge the beads out, and you end up with calcium and magnesium sitting in the bottom of your tube, very low concentrations in your lysate. You can take the lysate out, add back magnesium to activate the enzyme, and assay 30 or 40 samples, which is what I was getting from the schools each day, to actually measure the level of enzyme activity. At this point, Fred was a little peeved with me, because what I had told him was going to take 2 weeks to do had taken about 18 months. But we were finally ready to do the study, which we wanted to do initially. That is, we had an enzyme activity present in an easily accessible peripheral tissue, and we had what we thought was a relatively accurate assay for the enzyme. Now, at this time, we had started this study in the elementary schools, and by this time we were up to the junior high schools, which means that we were dealing with 13 to 15-year-old young adults, which is what I call them now that I have a 14-year-old daughter who keeps me honest about this. They are not children anymore, she tells me. And I show you this slide to make the following point. That is, that there is a, if this is the range of enzyme activity, there's a 4 to 5-fold range in enzyme activity in the boys, the girls, and the total population. And I show you one other thing here, which I don't expect you to believe at this point. And that is, this is not our nice bell-shaped Gaussian distribution. About 25% of the population looks like they're down here having a somewhat lower level of enzyme activity. Now, that was true in the girls and the boys and the whole population, but as I say, I wouldn't expect you to believe that, except I'm going to show it to you half a dozen times more, and hopefully I'll beat you into submission before we're done here. Now, we then advanced the study to the high school, and these are the ages of our high school youngsters. They're 16 to 18, and this is about, I don't think you can see the end, but this was about 350. It was about 250 in the other study. Once again, the same 4 to 5-fold range in enzyme activity, and the same 25% of the population approximately having relatively low enzyme activity. This activity, by the way, was not due, this range is not due to endogenous enzyme activators, inhibitors, or competing enzyme systems based on the addition of purified enzyme to samples across the range of activity samples. Now, because we were getting blood samples from so many of these youngsters, if their mothers and fathers would sign the consent form for one child, they generally let us take blood samples from several children in the family. So at long last, 600 samples and 2.5 years into the study, we could do the study we wanted to do originally. That is, we could see whether brothers and sisters are relatively alike with regard to the level of this enzyme activity. And here you see the sibling-sibling correlation for the 13 to 15-year-old youngsters. And you can see that although if you're used to looking at data from inbred rat strains, that it's not a nice straight line. There aren't many points here. There aren't many points here. And the correlation coefficient is about 0.6 and is highly significant. And if you look at the data from the high school youngsters, no points here or here. The correlation coefficient is about 0.5 again. This is a significant familial aggregation of the level of enzyme activity in the red blood cells of these people. Now, let me make the following point. If you're dealing with a biochemical variable that is regulated by polygenic inheritance, then one would expect that siblings who share on the average half their genes would have a correlation coefficient of about 0.5. Monozygotic, identical twins ought to have a correlation coefficient of 1.0. And indeed, studies done both here and done elsewhere have shown that if you look at monozygotic, identical twins, you get virtually a straight line with a correlation coefficient of about 0.96 to 0.98. Now, these sort of data don't prove heritability. These show familial aggregation. We can't separate out the effects of shared environment from those of inheritance from these sort of data. It's conceivable, for example, that all of the children with high COMT live in households where the parents say you will get good grades in school, and all of the children with low COMT live in households where the parents don't care whether they get good grades in school. Shared environment. But I can tell you that subsequent studies have demonstrated that the heritability estimated from these sort of data is very high for red cell COMT activity. Well, is that a big surprise? Well, it's a big surprise to pharmacologists, but of course it's not necessarily a big surprise when you stop and think about it. That is, you can see how tall I am. My wife is about 5 feet tall. I have a 9-year-old son. I have told him he will not grow up to be a professional football player in the United States because they have to be this tall and weigh 250 pounds. That's no surprise to me, and it's no surprise to any of you in this room. We know he picked the wrong parents to be a professional football player, but we know that the effects of inheritance on body size are due to a variety of genetic loci interacting. That is, this is polygenic inheritance. Well, that's fine, but from a biochemist's point of view, that sort of inheritance is very difficult to deal with because I don't have very much of a fighting chance to define the biochemical basis of the effects of inheritance in a polygenically inherited system. On top of that, the National Institutes of Health will want a progress report on my grant within about 6 months after giving it to me, and I'm sure I'll make no progress if I'm dealing with half a dozen or 10 or 12 different genetic loci. So the real question that a biochemically oriented type like me would want to ask is it possible that the effects of inheritance on this level of enzyme activity are due to a nice Mendelian inheritance, monogenic inheritance, autosomal recessive, autosomal codominant, sex-linked. And that possibility certainly arises. Here you see data from, I don't think you can see it, 577 subjects showing this group down here with the lowish level of enzyme activity. And what if this subgroup inherits the trait of low enzyme activity in a nice Mendelian fashion, if it's monogenic? That gives me a fighting chance to understand the biochemistry. And so I want everyone to be very clear about exactly what we did next. We set out to test the hypothesis that the trait of low activity, defined arbitrarily by drawing a line through the nadir of this frequency distribution histogram, is inherited in a Mendelian fashion by going back and getting blood samples from the fathers, mothers, brothers, and sisters of individual children who fell into this subgroup. This is just to help those of you who are somewhat removed from your genetic studies, as I was when we began doing these studies, to remember the sorts of things that can happen with monogenic inheritance, in this case by an autosomal recessive mechanism, in which if you have two heterozygotes mating, you get Mendel's famous 25% figure, which many of my physician friends seems to be the only thing they remember from their genetics course. If you have heterozygote-homozygote matings, you get 50%, and of course, if you have homozygote-homozygote matings for the trait, you get 100% of the offspring showing the trait. And I show you now the results of studies which we did when we obtained blood samples from 91% of living first-degree relatives in 48 families. Here, the black circles and squares represent individuals who fell into that low subgroup, and I show you these mainly to show you that we actually went out and got all these blood samples. And here you see some other pedigrees from those families. You'll notice if a black circle marries a black square, they have lots of little black circles and squares, and everyone likes to see that, and that's very uninformative information genetically. Not very informative, it's dramatic, but uninformative. What you're really much more interested in are these sorts of pedigrees where neither parent demonstrates the trait, and yet some of the offspring have the trait. Now, because there are equal numbers of boys and girls among the affected offspring here, this makes sex-linked inheritance relatively unlikely. What about the possibility of autosomal recessive inheritance? That is it possible that these people are heterozygotes for the trait and that they're passing it down in the fashion we just saw? Well, there's a problem with the analysis. Let me tell you what the problem is. Those of you who took fruit fly genetics as undergraduates remember that the way you made your heterozygote-heterozygote matings was to take a male heterozygote and a female heterozygote and put them in a milk bottle and cap it up, and they mated, and then you counted the offspring and you got Mendel's 25% figure. Well, how did we find these children? We found the children by virtue of finding an affected offspring. We found them through the offspring, not through the parents. What that means is, in families with one offspring, we're going to miss 3/4 of those families because of the ascertainment method we used. In families with 2 children, we'll miss 3/4 squared, or 9/16. We'll only find the families from here down. That's called the bias of ascertainment. And luckily for us, population geneticists over the years have come up with ways that help one correct for the bias of ascertainment, all having slightly different assumptions when we use these various means to correct for the bias of ascertainment. In all our presumed heterozygote matings, we had data that were compatible with Mendel's 25% figure, and in the presumed heterozygote homozygote matings, the data were compatible with his 50% figure. That is, the trait of low COMT activity in the erythrocyte is inherited in human beings as an autosomal recessive trait. And we can go further than that now on the basis of analysis of our data performed by Dr. Richard Spielman at the University of Pennsylvania, and say what we're really dealing with here is one genetic locus with 2 alleles, one for low and one for high enzyme activity. And about 25% of the population is homozygous for low, about 50% is heterozygous, and about 25% is homozygous for high. This is an autosomal, codominantly inherited characteristic in terms of regulation of the level of enzyme activity. I should point out that this has been shown more elegantly by studies which were performed by Dr. Floderis and Dr. Vetterberg here. And this is one of the few things in catecholamine studies on which there's general agreement. Virtually any other topic that you take up, you'll find that there's disagreement about. Well, at this point then, we said, how about that? The level of the erythrocyte enzyme activity is inherited in a nice Mendelian fashion, and that regulates a large part of the variance that we find in the population. Now, why did we do these studies? Well, we kind of wanted to know whether there was a biochemical basis for this difference in enzyme activity. And of course, the first thing which comes to mind is the possibility that you're dealing with different amino acid sequences, different proteins. And so we looked at a variety of properties of the COMT from genetically high and genetically low individuals. We looked at Km values for substrate, at the effects of inhibitors of the enzyme, at pH optima. They were all the same. One very sensitive way that one can look for differences in protein structure is to look at the thermal stability of the enzyme. And here you see in the solid line thermal stability studies performed on a genetically high sample, and in the dashed line thermal stability studies performed on a genetically low sample. This comes from me, this comes from the chairman of our department, and that's led to a great deal of speculation, psychiatrically speaking, which we won't go into at this point. But the important point is this, that the genetically low sample is more thermal labile than the genetically high sample, and this is a significant difference. Here we have heated these samples up at increasing temperatures for 15 minutes at each point. Well, those are only 2 people, and I wouldn't pay much attention to that, so we went back and we now took 3 samples, 3 from 3 different individuals who were genetically low and 3 different genetically high, and now we heated the samples up at 48 degrees centigrade, a temperature we picked from that previous experiment, for increasing times. And we found that the half-life of the enzyme at 48 degrees was about 12 minutes for the genetically low samples and was about 21 minutes for the genetically high samples, and these were statistically significant. But after all, that's only 6 people. So then I had Fred take a look at 316 samples, and he was now up to about 2,000 samples that he had assayed just from individual people for this study. And here you see once again that about 1/4 of this randomly selected population, this is a whole separate group of people, fell into this low enzyme activity subgroup. And we heated these samples up at 48 degrees centigrade for 15 minutes and saw how much of the activity remained. And you can see here what the activity looked like after heating. It's even more dramatic separation of the subgroup. Let me emphasize, there is no necessary reason why the thermal stability, that is the proportion of the enzyme inactivated by heating, ought to be related to the genetically determined basal level of enzyme activity. Those 2 things don't have to be related at all. But when we looked at the proportion of activity remaining expressed as a fraction, as a heated over control ratio, you can see that the subgroup here of individuals who we already knew on the basis of our genetic studies inherited this trait of low enzyme activity had a more thermal labile form of the enzyme. And I'm showing you all the data points here. No nice little bar graphs with standard errors. You're seeing all the data. Then did the remainder of the population. If you like little bar graphs, I can show those to you, and because the n's are so large, the standard error of the means are the sorts of things that pharmacologists are not used to dealing with, and of course it's highly significant. What this indicates is that at least with regard to this one biochemical characteristic, there's a dramatic difference between people who genetically have low levels of enzyme activity, which we know are inherited. And that would enable you to say that probably we are dealing with a structural gene variant here, although we cannot rule out the possibility that this locus, COMT, that we're looking at, represents a genetically determined variation in post-translational biochemical modification of the protein. Now, this is a balanced polymorphism, which means that at least in our white Northern European population in Rochester, Minnesota, the gene frequencies are about equal for these two different alleles that we're looking at, and that means 25% are homozygous highs, 25% homozygous lows, and 50% heterozygotes. And we could stand around, if we were population geneticists, and argue about why this polymorphism is maintained in the population and what the evolutionary pressure might be, but we won't go into those sorts of controversial issues here. So, now about that, those data were published in 1977, and Fred was getting tired of doing the assays. We were now into that 2-week study. We were about 4 years into the 2-week study, and we had demonstrated that not only is COMT present in an accessible tissue and developed an accurate assay, and that there's a 4- to 5-fold population variance, and that a large part of that variance is due to genetic factors, and particularly this one locus with 2 alleles that was regulating the variance. But after all, we've only talked about the red blood cell, and we started out somewhat interested in what this might mean functionally. Does that have anything to do with what's going on in other tissues? We couldn't measure the enzyme activity in autopsy samples because the enzyme died very rapidly. But what we could do, since we're one of the busier surgical centers in the United States, is go to the operating room and get tissue from people who were having clinically indicated operations, have it dropped directly onto dry ice, walk over to the laboratory and assay it, and also assay the red blood cell COMT activity, which is genetically determined. And here you see human lung samples, which we obtained courtesy of the American Tobacco Company, because most of these people were having their lungs removed because of carcinomas of the lung. And you can see that the genetically determined level of red blood cell enzyme activity correlated significantly with the level of activity found in the human lung. Once again, relatively few points here and here. Well, with that kind of scatter, I'd prefer to see that in some other organs. And so we were also able to get kidney samples from patients who were having nephrectomies performed. Once again, the whole organ was removed because they had tumors. We went to the opposite pole of the kidney and looked at the COMT activity. And here you see the correlation coefficient is 0.8. Once again, highly significant. And in a study which we have just sent off, which has just been accepted and will be published in Biochemical Genetics later this year, we've also isolated human lymphocytes free of platelets and free of red blood cells, and determined whether the lymphocyte enzyme activity is correlated with the genetically determined red cell activity, and whether you express the lymphocyte activity per milligram protein or per lymphocyte, you find that there is a highly significant correlation. It appears that if you stick a needle in somebody's arm and draw a blood sample and look at their genetically determined level of COMT activity in the red cell, you can make some predictions about what the level of enzyme activity is in their lung, in their kidney, and in their lymphocytes. Now, I can't tell you anything about the brain yet because we still don't have an accurate way of looking at this problem in human brain. And remember, I said we were also interested in functional significance. And these studies that I've just shown you, and we started in 1973, are in press. So you can see the study has gone on and on and on. Well, let's see whether there's any evidence that this genetic variation has any functional significance. Let's look again at this drug, L-DOPA. One of the major metabolic pathways for L-DOPA involves catechol-methyltransferase. And here we have the formation of 3-O-methyl-DOPA by COMT. In studies which we did not do, but which were performed by Dr. Douglas Riley and Ravira Kalimlim at the University of Rochester, and this is Rochester, New York, not Rochester, Minnesota, and actually it's hard to believe, but I had a postdoctoral fellow come from Munich, and he sent me a telegram and said, arriving at Rochester Airport on Allegheny Airlines flight so-and-so, and the problem is that Allegheny Airlines does not fly to Rochester, Minnesota, and this poor fellow landed in Rochester, New York, and it wasn't until he got there that he found out he was 1,200 miles away from where he was supposed to be. I don't know who his travel agent was in Munich, but that actually happened. He's now safely working in the lab on phenyl sulfotransferase, which I'll tell you about in a minute. But this work was done in Rochester, New York, and what you can see here is the following: that the level of of erythrocyte COMT, which we now know is genetically determined, correlates directly with the proportion of L-DOPA converted to the 3-O-methyl DOPA metabolite in patients with Parkinson's disease being treated with the drug. That is, this genetic variation, at least in a pharmacogenetic sense, has functional implications. And not only that, there's a direct relationship with resistance to the drug by patients who are, if you will, rapid methylators, at least according to this report by Douglas Riley and Revere Cullenlam. I can't give you any information yet about any possible functional significance with regard to the metabolism of endogenous catecholamines. But at this point, we had found not only that COMT is present in accessible tissue, developed an accurate assay, looked at the population variance, determined that that is primarily genetic in basis, found that what we were looking at in the red cell at least reflected what was going on in 3 other tissues or organs, and it has been shown that it has some functional significance, at least pharmacologically, but we don't want to ignore the effects of other enzymes. Well, what am I talking about here? Well, if we go back and look at dopamine, remember that we pointed out that not only can dopamine be metabolized by COMT, but by these other 2 enzymes, phenylsulfotransferase and monoamine oxidase. Now, 25% of the population of Rochester, Minnesota, which is in large part Scandinavian, and we can say about 25% of the population in at least certain parts of Sweden, have genetically determined low levels of COMT. Even in the wintertime, I don't think that 25% of the population of Rochester, Minnesota is either schizophrenic nor psychotically depressed. So we don't really think that that alone is going to result in psychiatric illness, assuming that what we measure in the erythrocyte reflects what's going on in the brain, and that remains to be proven. But is it conceivable? We know, for example, that monoamine oxidase, which occurs in 2 forms, monoamine oxidase A and monoamine oxidase B. The A form can be measured in fibroblasts, the B form in the human platelet. Both of them appear to be regulated in man, in large part by genetic factors. We know that MAO is regulated genetically, at least in these systems. The system has not been worked out as carefully as with COMT. Is it conceivable that there are people walking around who either on a genetic basis or on the basis of some environmental factor might have low levels of COMT, low levels of MAO, and low levels of this other enzyme, phenylsulfotransferase? Might those people behave somewhat differently than people who had genetically high levels of all of these enzymes? Well, in order to begin to try and answer those questions, we're going to have to go through the same procedure for each of these enzymes in turn. Can phenylsulfotransferase be measured in an easily obtained peripheral tissue? I think we need somebody to stick on the next tray. Leonard couldn't believe that I could actually talk fast enough to have more than 1 tray's worth of slides. And it should just slide right in, and if the slides are the wrong way, it's entirely my fault, no one else's. All right, we'll go backwards here. There we go. Well, we set out to find out whether phenylsulfotransferase could be measured in an easily obtained peripheral tissue. This shows you one example of a reaction catalyzed by phenylsulfotransferase. Here you have MHPG, the norepinephrine metabolite formed in the brain, which psychiatrists are so fond of measuring, and it shows that MHPG can be sulfated in the presence of, in this case, a sulfate donor, phosphoadenosine phosphosulfate, to form a sulfated conjugate, which, if we use radioactively labeled sulfur, we can then use as the basis of an assay procedure. And the way the assay works is to just take the sulfate donor and the substrate MHPG and shake them up together with whatever tissue you're looking at, incubate them, precipitate with barium the sulfate donor, centrifuge out the precipitate, take the supernatant off, and measure the radioactivity in a liquid scintillation counter. And that sounds very easy. And in 1977, Dr. Robert J. Anderson, who is of Swedish origin, entered our laboratory, and he was going to be a postdoctoral fellow. And he was of a clinical background, and he wanted a project, and I said, let's see whether we can measure phenylsulfotransferase in the human erythrocyte or in any blood elements. And his first day in the lab, he took the radioactive donor, PAPS, and he shook it up with MHPG and red blood cell lysates, and took it over to a scintillation counter, and his active samples had thousands of counts in them, and the blank samples had very low counts, and he said, boy, science is easy. And then he tried to get an accurate assay as opposed to having an assay, and those are two quite different things. And about a year and a half later, he had found that there are potent endogenous inhibitors of that enzyme found in red cells and platelets, both of which contain the enzyme activity, and had worked like a dog and learned a lot more biochemistry than he cared to find ways to get rid of those inhibitors. And we finally had an accurate assay using MHPG as a substrate to measure platelet phenol sulfotransferase activity. And what we found when we did this was, number one, that there was about a 5- to 6-fold variation in the population in the platelet enzyme activity. That is, there is a wide population variance, just about the same as for COMT. Number two, we were using MHPG as a substrate, but after all, we were also interested in such things as dopamine, which is a substrate for this enzyme. We were interested in 5-hydroxytryptamine, serotonin, which is also a substrate for this enzyme, and believe it or not, even paracetamol, as you call it here, or acetaminophen, or Tylenol, which is its trade name in the United States, a widely used over-the-counter analgesic, is metabolized by the same enzyme. So I can justify doing these studies to my chairman of a pharmacology department, even if it has nothing to do with psychiatry, because it has to do with drug metabolism too. And what we did was take samples of platelets from, in this case, 16 individuals, and use both dopamine and MHPG as substrates, and you can see that it appears that we're measuring the same enzyme activity. The correlation coefficient is 0.82. And when we look at serotonin as a substrate for the platelet enzyme, you can see a high degree of correlation when we use serotonin. These are 20 samples from 20 individual patients, and you can see this range of enzyme activity, and you can also see that both serotonin and MHPG appear to be measuring the same enzyme activity. The same is true of acetaminophen or paracetamol. So we now have, oh, and in addition, Dr. Anderson obtained kidney samples from patients undergoing nephrectomy for clinically indicated reasons. It took him 8 months, even at our very busy surgical center, to get these samples from 20 patients. The correlation coefficient is only 0.5, but this is significant. It looks like what we're measuring in terms of relative activity in the platelet may reflect what's going on at least one other organ, the human kidney, and the correlation coefficient for human gut samples obtained by having people swallow biopsy tubes was 0.7. So it may reflect what's going on both in the intestine, which is where this enzyme is also located, and in the human kidney. And if this sequence of events looks familiar to you, it ought to, because this one began in 1977, and we now have an accurate assay, and we know that we can measure the enzyme in a peripheral tissue, and we're looking at population variance, and you don't need very much imagination to know what we're going to do next. And I'm giving you a preview because we're just beginning these studies. But Dr. Anderson has now moved on, having published several papers, about half a dozen papers, and is an independent investigator at the University of Illinois. The year and a half didn't scare him off, I'm happy to say. Well, what's the point of all of this? Because what I've really done is give you a progress report. The point of all of this is that maybe this is one way that we can begin to gain in human beings, as opposed to experimental animals, and we learn a great deal from experimental animals, begin to gain some insight into the regulation of variation of one part of the function of one important neurotransmitter system. Now, frankly, I think over the long haul, these data will have clear implications with regard to pharmacology, individual variation in the metabolism of drugs, and that's already been shown for the COMT genetic polymorphism. Perhaps they'll have implications with regard to neuropsychiatric disease, which is the thing we're all interested in. That still remains to be demonstrated. It remains to be tested in the future. It's taken a long time to get to the point that we could even say that. Also, I would like to think that this one very small part of the nervous system, that this sort of an approach, will be the sort of an approach which will help us understand other neurotransmitter systems. And of course, enkephalin is a word with a great deal of sex appeal today. Enkephalins are just substances. They are, in this case, small polypeptides, but they are degraded by enkephalinases. Are there inherited variations in enkephalinases? Well, I'm sure that somebody will someday find out about that. Are there inherited variations in the ability to synthesize some of these compounds? What about serotonin? Well, there are all sorts of neurotransmitter systems, and I'm sure that before we're done, we'll learn a great deal more of this same sort of information with regard to inherited regulation of the enzymes that are involved in both the biosynthesis and the metabolism of all of the neurotransmitter systems that we presently know about, and we're only beginning to scratch the surface today. And unfortunately, or fortunately I think, some of this is eventually going to have clinical implications, both in terms of drug metabolism and in terms of the metabolism of endogenous neurotransmitters and behavioral differences among individual human beings. Now, there are two things I want to do before I stop. First of all, I want to point out that there are people like me who run around the country and the world talking about studies, and there are people who do studies. And as the years pass, I do fewer and fewer, unfortunately. And these people have all been involved at one time or another in these COMT studies. Mr. Raymond, several years ago, decided that he had done enough COMT assays and is now a full-time computer programmer. But all of these people have been involved in doing the COMT studies. The one other thing I'd want to do is that as I do travel around, I find, and I don't know whether we can turn these lights down or not, but I find that there is some interest in where the studies were done. And I therefore will show you these slides showing you pictures of the Mayo Clinic in Rochester, Minnesota. Rochester, Minnesota is a very small city. It is a city of about 70,000 people located in the cornfields in the Middle West. You can see the limits of the entire city here. And all of this complex in the downtown area of Rochester is the Mayo Clinic. Here's the St. Mary's Hospital, which was built in the 1880s. It has about 1,200 hospital beds after a tornado destroyed one end of the town. This is the Rochester Methodist Hospital, which has about 1,000 beds. This is the present Mayo Clinic building. It's a 20-story building, research building. All of these buildings down here are part of the Mayo Clinic complex. There are in this city of 70,000 people. And here you see the Mayo Clinic building, the Rochester Methodist Hospital, the St. Mary's Hospital. This is the research building where I work. Sort of peeking around the corner is the original Mayo Clinic. There are 2,000 physicians in a city of 70,000 people, about 2,200 hospital beds. The current estimates are that this 20-story building will have to build a building twice that size by 1990, a 40-story building in addition to this at our current rate of growth in order to meet the patient care demands. It clearly is the largest clinic in the world. And I should probably make the point, because people seem puzzled by this, this is the building in which that research was done, which I've just described, that this is not a group of physicians banded together for individual profit, but for the last 70 years this has been a non-profit medical foundation. All the profits from this clinical operation go back directly into biomedical research and education, which made it possible to build this building and to do the studies I've just talked about. I want to thank you very much for your patience. I'll be happy to try and answer any questions that you have. It's been a real pleasure to be here with you today. Thank you. Anyone have any questions? I can't believe it was that intimidating. Yes?
Do you have any idea of the clinical significance of your findings? I mean, preliminary.
The only clear-cut clinical implication to come out of it, although I'm a clinician, you see, the only clear-cut clinical implication to come out of this to this point is that this genetic polymorphism for COMT has direct relevance with regard to resistance to L-DOPA therapy in Parkinson's patients. Now, I think you could probably surmise that it may well have significance in terms of individual variations of the metabolism of alpha-methyl-DOPA or Aldomet, and perhaps of other catechol drugs such as isoproterenol, but those are speculations. I once was asked by a resident at the University of Michigan, after I talked about some of these data, he says, well, what if you spend 10 years and you get to the end of that and you find out that you've described this nice genetic polymorphism and it has no clinical significance? What will you do? And I said, I'll go on to another enzyme. What do you think I'm going to do? I make you no promises about clinical significance. I wish I had the answer to your question. Obviously, it's taken us years to get to the point that we can begin to test those sorts of hypotheses. And I should point out, I think in all fairness, I've shown you nothing but data on humans. And that's my primary interest. But a great deal can be learned from experiments using inbred animals and experiments done with purified enzymes in the laboratory. We do a certain amount of that also, and a great many other investigators are doing that sort of work. And we're learning a great deal about catecholamine biochemical genetics, which we hope we can then apply to man. But a decade ago, If one had wanted to give a lecture about catecholamine biochemical genetics, it would have been one of the quickest lectures in the world because there was nothing known. So when you take that sort of a perspective, and I was actually working with catecholamines a decade ago, it's hard for me to believe that, although easy for my children to. When you take that sort of perspective, what you can see is that, that we have come some ways. Not as far as you'd like and not as far as I'd like. Yes?
I was thinking, there was an article published a while ago where somebody had compared the drug metabolism using two populations of rats. I can't remember the drug and I can't remember the author. Anyhow, they had one inbred population of rats, so they had this beautiful curve going right down. And then they'd also gone out and caught rats that were living in the same city as the authors and put these rats into cages and done the same thing, and they found tremendous variability within the individual rats. And so these people were asking if actually one could, just by mistake, be using a rat for drug assays who was way out at, you know, it's quite far from the mean.
Well,
I thought that probably this would account for some of the differences that we see in patients when we give the same patient the same dose of the drug and get to very different results.
I couldn't have said it better myself. That's exactly the point. You have just summarized the whole point. We are not an inbred strain. And I find it amusing. We've done biochemical genetic studies of COMT in inbred strains of rats, and have found inbred strains of rats with dramatically different levels of COMT, and you breed them and it segregates as a nice monogenic characteristic, just as you might expect that it would do. And obviously that's all very nice, and it's a nice model for us to work with because we can grind up the brains of rats as opposed to human beings. So that's what we're doing in the laboratory with the rats. But I think the point that many of my pharmacology colleagues, and I'm in the Department of Pharmacology, many of my pharmacology colleagues miss is that they use for their experiments inbred strains. They don't like the variants from animal to animal. That's not what they're interested in. What have we been talking about here? We've been talking about the variants found in a mongrel outbred human population. And we call those things polymorphisms when we look at enzymes. And some of us think that they might have clinical significance. Now clearly they do. You already know about acetylator status and the variation that that induces in terms of isoniazide, hydralazine, metabolism, right? And that has clinical significance. In hydralazine, it has clinical significance with regard to the occurrence of a lupus erythematosus syndrome, and with isoniazide with regard to the occurrence of the isoniazide hepatitis. And yet we go on year after year using the same doses to treat everybody as if we were dealing with inbred rats. So I couldn't have made the point better. Thank you very much. I did not pay her before to make that comment. If there aren't any other questions or comments, thank you very much. It really has been a great pleasure to be here with you.
Thank you. I think the discussion can continue upstairs. We will have some refreshments there. We will have discussion in small group. Thank you again for coming all this way.